Hi everyone, I am Dr. C.S. Kulkarni, MD, and I am going to teach medical biochemistry. Before I start the topic, please like, share, and if you are new to the channel, please subscribe it so that the next video will be intimated to you. In the last session, we ended up with phospholipids. In today's session, we will start with sphingomyelins. Then we will study glycosphingolipids, cerebrosides, sulfatides, and gangliosides. And lastly, we have lipid storage diseases. Good morning, everyone. Let us begin the topic. Synthesis of sphingomyelin. Ceramide plus CDP choline gives sphingomyelin plus CMP. Ceramide reacts with CDP choline to form sphingomyelin. The reaction being catalyzed by the enzyme CDP choline ceramide phosphocholine transferase. Coming to synthesis of glycosphingolipids. These carbohydrate containing lipids are synthesized by transfer of an active glycosyl or hexosamine residue from the UDP derivative. Coming to synthesis of cerebrosides. The most common ones are glucocerebroside and galactocerebroside. The transfer of 1 beta glycosidic bond between the C1 hexose and C1 of ceramide is catalyzed by specific glycosyl transferases. Galactocerebroside is an important component of brain lipids. Synthesis of sulfatides. Cerebroside sulfatides are sulfur containing esters of cerebrosides, and the major sulfolipid of the brain is galactocerebroside 3 sulforate. PAPS or phosphoadenosine phosphosulfate or active sulfate formed from sulfur containing amino acids. The enzyme is a sulfotransferase. Coming to synthesis of gangliosides. This group of glycosphingolipids contains one or more sialic acid residues. They are present in high concentrations in the CNS and also on surface of membranes. The gangliosides of brain are GM1, GM2 and GM3. For the ganglioside synthesis, the active form of NANA, that is N-acetyl neuraminic acid, used is its CMP derivative. The NANA attached to the hydroxyl group of its second carbon and to the third hydroxyl group of sugars. The specific ganglioside on intestinal mucosal cells bind to the B subunit of the cholera toxin when the A subunit enters the cell. It keeps the level of cellular CMP raised by decreasing GATPase activity by the G protein. Here we can see synthesis of gangliosides. Ceramide glucose galactose plus CMP NANA forms ceramide glucose NANA with the removal of CMP. This further forms GM3. Then this ceramide glucose galactose NANA forms ceramide glucose galactose NANA and galactose neuraminic acid in the presence of UDP N-acetyl galactosamine and the removal of UDP, and this further forms GM2. Gangliosides also act as receptors for other toxins like tetanus toxin and toxins of viral pathogens. Gangliosides contribute to stability of paranodal junctions and ion channel clusters in myelinated nerve fibers. Autoantibodies to GM1 and GD1A disrupt the lipid rafts, the paranodal or nodal structure, and ion channel clusters in peripheral motor nerves. Coming to the next point, lipid storage diseases. In the diagram, you can see generalized gangliosidosis due to the absence of the enzyme beta-galactosidase, Santos disease due to the absence of enzyme hexosaminidase A and B, Tay-Sachs disease due to the absence of enzyme hexosaminidase A, Fabry's disease due to the absence of enzyme alpha-galactosidase, lactosyl ceramidosis due to the absence of enzyme beta-galactosidase. Gaucher's disease due to the absence of enzyme beta-glucosidase. Metachromatic leukodystrophy due to the absence of enzyme aryl sulfatase. Krabbe's leukodystrophy due to the absence of enzyme beta-galactosidase. To Gaucher's disease, as the enzyme defect is beta-glucosidase. Enzyme accumulated is glucocerebroside. There are three types of Gaucher's disease: adult, infantile, and juvenile. Hepatosplenomegaly, erosion of bone, and moderate anemia are seen. Niemann-Pick disease. Enzyme defect is sphingomyelin. Lipid accumulated is sphingomyelin. Severe CNS damage, mental retardation, hepatosplenomegaly are seen. Cherry red spot in macula is seen, and death occurs by 2 years of age. Krabbe's leukodystrophy. Enzyme defect is beta-galactosidase. Lipid accumulate is galactocerebroside. Severe mental retardation, total absence of myelin in CNS, and globose bodies in white matter are seen. Metachromatic leukodystrophy. Enzyme defect is sulfatide sulfatase. The lipid accumulated is sulfogalactocerebroside. It accumulates in most tissues. Neurological deficit, difficulty in speech, and optic atrophy are seen. Demyelination is also seen. The enzyme defect is alpha galactosidase. Lipid accumulate is ceramide trihexoside. Kidney is a site of accumulation. Progressive renal failure is seen. Death by 5 years of age is seen, and purplish papules appear, and X-linked inheritance is seen. Tay-Sachs disease. It is the enzyme defect is hexosaminidase A. Lipid accumulated is ganglioside. Incidence is 1 in 6,000 births. Mental retardation, cherry red spot in the macula, and progressive deterioration is seen. Death by 3 to 4 years of child is seen. Generalized gangliosidosis. Enzyme defect is beta-galactosidase. Lipid accumulated is ganglioside. Mental retardation, hepatomegaly, skeletal deformities are seen. Foam cells are seen in bone marrow. Cherry red spot in the retina is seen. Lactosyl ceramidosis. Enzyme defect is beta-galactosidase. Lipid accumulated is lactosyl ceramide. Mainly CNS and reticuloendothelial system are affected. Sandhoff's disease. Enzyme defect is hexosaminidase A and B. Lipid accumulated is globoside. Neurological deficit and mental retardation are seen. Multiple sclerosis. It is a demyelinating disease. Phospholipids, that is, ethanolamine plasmalogen and sphingomyelids, are lost from white matter of the central nervous system. The cerebrospinal fluid contains increased quantity of phospholipids. Coming to the lipid storage diseases, they form a group of lysosomal storage diseases. The sphingolipids are normally catabolized by a series of bond-specific lysosomal hydrolyses like alpha-beta glucosidases, galactosidase, neuraminidase, hexosaminidase, and aryl sulfatase. The disease results from failure of breakdown of a particular sphingolipid due to the deficiency of a single enzyme. The children afflicted by these diseases are severely retarded mentally and seldom survive for long. These diseases can be diagnosed prenatally by amniocentesis and culture of amniotic fluid cells. Since the children born with these diseases will have severe mental deficits, the pregnancy may be terminated. Replacement of severe of enzyme has been tried in culture disease with limited success. The common features of lipid storage diseases include only one type of sphingolipid accumulates, rate of synthesis of lipid is normal but degradation is affected, and the extent of enzyme deficiency is the same in all tissues. Here ends our session. Kindly like, share and subscribe the channel. Thank you.